Am I on?
Yep. Thank you so much for sharing your personal perspective as well as your professional. All right. And Rob will be around for questions as well. All right. Now we'll hear from Toby.
Thanks again. And so I'm going to take a slightly different perspective of tell you about my experience in another disease. And it's similar to what I've said yesterday, but I'm going to try to slant it a bit differently. I mean, fundamentally, my perspective comes to this as a researcher and a neuromuscular neurologist. In a prior life, when I was a practicing clinician, I used to take care of people with CMT. More recently, I've been working on another disease, ALS, also a neuromuscular disease, also a disease of motor neurons. And so I think fundamentally there are lots of lessons learned. But if I could start off with what do I think is the take home for everyone in this room, but particularly those with you with CMT, is that if we get the science right in this disease, we can bring forward, all of us together, truly meaningful therapies that change the lives of the people with CMT. And the example I'm going to offer you today is an experience that did that in ALS, or is doing that in ALS. And then I'll try to make some points about what are the tools and pieces that we can learn from that experience and how we can apply that to CMT. This just makes the point that the process of developing a drug, it can be long and difficult, and there are always bumps along the way. And in some ways, those bumps actually become quite informative. You'll hear later in this story that a biomarker, in this case neurofilament, was crucial to the success of this molecule I'm going to talk about, Ferguson. If the initial trial had been successful and we sort of sailed through, we wouldn't have learned all we learned about neurofilament. And so, in some ways, I think, Rob, said it nicely. You take action and then you take the lessons learned and you keep moving forward. So just a couple words about ALS. So ALS is a disease of the peripheral and central nervous system, really a disease of motor neurons. The key point about it is that you lose your strength over time, you lose your function. In that way, it's pretty similar to CMT. But you also lose, in this case, you lose your ability to breathe, and most people die in about 3 to 5 years. This talk is about one form of ALS called SOD1 ALS, and the key point here is that SOD1 ALS is exceedingly rare. In the US, there are 300 to 600 people with this disease. So what's the key point here for this broader community? Is even when the diseases are incredibly rare, you can effectively develop therapies in those diseases. And that's an important point. And that only happens with the coincidence of clinicians, patients, researchers across the spectrum from science to industry. You've heard some nice preface comments from Bruce and Rob about sort of the modalities and how you approach this. In this case, this molecule that we're going to talk about is called Ferguson. It's an antisense oligonucleotide, which means that it gets into the cell, in this case, motor neurons or other cells, binds to a bit of mRNA and leads to the destruction of that mRNA. And that's important because we understand the basic science of SOD1 ALS to be that the mutation in SOD1 is somehow toxic to those motor neurons and causes them to die. And so the fundamental therapeutic hypothesis is you want to reduce those levels of that toxic protein. And so in this case, antisense is a great way to approach that. I guess another point I'd make, and I'll make it again at the end of the talk, is that really understanding the mechanism of the disease is absolutely critical to your chances at succeeding. Knowing the genetics is a key first step, and a key second step is, okay, how does that genetics influence this disease? I think CMT, this community is fortunate, and you have a number of defined genetic components of your disease that really help us understand the basics of the disease. And that's really the substrate that enables us to develop highly effective therapies. And that inflection point you saw in Rob's slides really, in my mind, point to a coming inflection in therapies. This just highlights that there were a number of trials that contributed to this overall discussion. I'm going to focus on the main trial that tried to ask the question, does the drug work? And by work, I mean, does it provide a change in functioning for people with ALS? The trial was short, had a short placebo-controlled period of about 6 months, which is what you see is called Valor here. And we were trying to find ways to enrich for faster progressing patients in that context. And we had some tools to do that. A fundamental point is they didn't work, and you'll see that becomes important later. But the key things we measured in this trial were the ALS Functional Rating Scale, which is a clinical scale. In general, what I'd say about clinical scales, and this is true across neurology in my experience, CMT, ALS, Parkinson's, clinical scales are often difficult, often variable, and so sometimes they're hard to interpret. And so biomarkers, which we also measure in this trial, neurofilament and SOD1, become really crucial. So this was the initial result in the study, and in 6 months, you can see at the end, there was very little difference between the treatment and the control. So the initial interpretation of this data would be this drug doesn't work at all. So how do we move this forward with this result? So this is the biomarker data, and I highlighted previously that biomarkers were really critical to the success of this molecule. The biomarker on the left is just SOD1, and so I told you already that the goal of the study was to knock down SOD1 and see what happens. And so the first question you need to answer in drug development is your molecule doing what you think it should be doing? So in this case, we show in the spinal fluid of people with SOD1 ALS who've gotten this drug that the SOD1 is reduced. So that's a really key critical step. So the opportunity you have in CMT is to ask this question for each trial that comes forward. For example, in the CMT1A trial, the question would be, okay, have we reduced PMP22 to the level we think we need to? So measurement of PMP22, for example, in that kind of trial is going to be critical. The next biomarker we looked at was neurofilament. I'll get into a bit more detail in a moment, but essentially neurofilament is a marker, it's found in axons and neurons, and it's a marker of axon injury. It's not specific, so it happens to be elevated in many diseases, but what we saw here was a very clear reduction. So seeing the clinical data didn't look supportive, but biochemical data was really promising. So just to level set a bit more about neurofilament, I've already told you that it's found in axons, it's shed nonspecifically after injury, and I think the important point to make is also it can be easily measured. You can measure it in spinal fluid, but you actually can, there's nice blood tests for it. And so very simply, you can take a simple blood measure and understand what the levels are and do those levels change over time. This just highlights that most of the research in ALS and neurofilament was a relatively recent phenomena. The trials that I highlighted previously had started in the 2015. And so really only during the course of these trials was neurofilament data really being generated broadly in the ALS community. And this was done across many groups, many parts of the world. But that community contribution to this was critical to our understanding of the behavior of neurofilament in people with ALS. This just highlights the key basic data, and that is the higher levels of neurofilament, the worse you do. On these slides, you see these are measurements of clinical function, like the ALS Functional Rating Scale, but they're basically how people are moving, breathing, eating, among other things. On the other hand, this slide actually shows if someone's not doing as well, you expect they may not live as long, and these data support that. So the higher your levels, the shorter your survival. And so we have two pieces of key information that your levels of neurofilament predict how you do and feel and function, and they predict how long you survive. And so I think in pulling lessons through to thoughts about biomarkers in CMT, drawing those relationships to how biomarkers we measure, what those implications for our people in their daily lives, is a crucial connection. And you'll see why as the development of this drug, that becomes a crucial question the FDA becomes quite interested in. This just highlights that I told you already that we had wanted to try to select for faster patients in that first 6 months of the trial. We used some tools. We used ALS progression clinically, and we used mutation rate, and those weren't particularly good it just turns out neurofilament was better. So a key lesson learned from this study was that in trying to control for the clinical heterogeneity that you have in neurologic disease, the best tool we had was actually neurofilament. So these are I sort of, when you think about biomarkers, they have different uses at different times in trials, and we've highlighted 3 here for neurofilament. One is you can see a rise of neurofilament early in disease, and that's on the far left. So that tells you about the onset of damage to your nervous system. In the red box, I've highlighted the 2 examples we've shown here. One, we've told you that your levels are predictive of your clinical course. Therefore, that could be a tool when you're thinking about your control group and your experimental group to control for your heterogeneity. That becomes a very important practical tool. The other one, you can ask yourself, in this case, because the neurofilament levels are elevated, if you see a reduction, that means your molecule could be working. So this is sort of where Biogen, who developed this drug, found themselves. They had supportive clinical data. They had a clear biomarker response. And so they then approached the FDA and said, can we get this drug approved? And really what they asked for was an approval based on an accelerated approval. And what that means is a mechanism to basically get a drug approved when there still remains some uncertainty about the effectiveness of the drug, but a clear effect on a biomarker. And so there are a couple key phrases. One, is there a serious need, or is it a serious condition? In ALS, in CMT, in almost all neurologic diseases, that's a very clear and simple case. What's the severity, rarity, or prevalence of the condition and the availability of alternative treatments? Unfortunately, in lots of diseases like CMT and other neurologic diseases, there's a clear unmet need for directed therapies. So that's straightforward. So the real key question then becomes, whatever your surrogate is it likely to predict benefit in people? And that really comes down to what is the plausibility of that relationship of the biologic effect to the clinical effect, and is that relationship quantitative? And I've made a clear point about trying to highlight that relationship of neurofilament to the eventual clinical outcome. And it's that data set developed by the entire community that enables that discussion with the FDA. And so when we went and had these discussions with the FDA, it really was about those relationships, both quantitative and qualitative, that supported that the molecule was likely to work. The FDA didn't take our word for it. They reanalyzed the literature themselves and confirmed the responses that others had seen as well. And so in totality, where were we? I haven't shown you already, but there's also quite a strong biological plausibility for the molecule as a whole. We knew preclinically that you could treat the SOD1 mice, which are the mice with the disease model, and you saw neurofilament reductions. In an earlier study, in a phase 1/2 study, we saw consistent changes in neurofilament. And in a later study, we also saw the one I described already, we saw these changes. So the effect was very robust throughout every situation in which the molecule was investigated. We also looked at the molecule in different circumstances. We looked at it in plasma, and we looked at it in 2 different types of neurofilament, neurofilament light and neurofilament heavy. In all cases, the response was consistent. And then, so we also looked at the longer-term clinical effect. And here what you can see again is the neurofilament on the top, but on the bottom you can see, in the case of the ALS functional rating scale, slow vital capacity, which is a measurement of breathing and strength, in all cases, those people who in green were those who got treatment earlier appear to do better. And so fundamentally this data, it's this clinical data plus all the neurofilament data I've been through, that in and of itself together supported the approval of tofersen under an accelerated approval mechanism. What that means is the FDA agreed the drug was likely working. based on all this data, both clinical and neurofilament. They also said, okay, this is good, we'll allow you to give this to patients, but you have to go further confirm the efficacy in another trial, and that study remains ongoing. They're now starting to be real-world experience with tofersen, and here what you can see is this is a study in Germany of about 16 ALS patients who've gotten tofersen, and you can see in a measure of the ALS functional rating scale of vital capacity, if the blue line is the baseline when they enter the study, you can see that the means have increased throughout the course of the study. And so you've heard this a couple times based on the inherent capacity of the peripheral nervous system, that improvement may be possible. So this is a living example in a group of ALS patients that over time they've actually done better. So I think if you ask my perspective on, I think if you target your science appropriately, the peripheral nervous system and the right stage of disease, I think there are inherent capacities in the peripheral nervous system for potential improvement. This is one example. Dr. Bonema's talk yesterday is another example. Dr. Carter mentioned sort of the endogenous capacity of Schwann cells to support repair and regeneration. So I think these examples highlight that if we get things right, we really can change the course of these diseases. So switching focus to a little bit more explicitly on CMT, the diagram on the left just highlights sort of a current assessment, and you've seen this in various forms throughout this discussion, of the genetic state of CMT. And the key point I want to make here is just that knowledge is a prerequisite to understanding disease in detail and then making therapies. And CMT is fortunate in the sense that we have this accumulated knowledge. The slide on the right this is a preclinical model of CMT1A. Just one example of a disease which we have genetic understanding in CMT. The point I wanted to make is that in this case, they've knocked down CMT with an ASO. The exact modality is not critical, but the point is they've sort of addressed the underlying disease mechanism. And in this context, they've measured compound motor action potential, and they've measured grip strength, just two measures of both electrical and function in a mouse. And there you saw repair. So I've told you now a couple times about the potential for repair, and I think this is just a preclinical example of that possibility. I think you've seen some versions of this already, but fundamentally, getting things to the right place is a critical and practical issue. Getting them to the motor neurons, getting them to the Schwann cells, these are things we have to address fundamentally and ask ourselves, in each study, are we getting stuff to the right place? That I highlighted sort of measurement of SOD1, that's what we call target engagement. That's really that critical first step of asking yourself, are things getting to the right place? Because if you don't know that, it's hard to interpret the other data downstream. So this is just sort of my summary. So I think fundamentally there are quite a few similarities and there are a few differences across CMT and ALS. But I think fundamentally, we have deep understanding of the disease. That provides an opportunity to make meaningful therapies. The trials themselves aren't always straightforward, and getting that experience, getting things in the clinic, getting multiple goals is critical. When we go through those experiences, we'll have wins and we'll have failures, but learning from those failures, incorporating different tools like biomarkers is absolutely crucial. I've given you an example here where this drug wouldn't have been approved without a biomarker. I've also highlighted it. I'm optimistic about the state of the science in this field and the potential for meaningful therapies, and I've given you some practical examples of that. I think fundamentally in ALS, we've de-risked a little bit of getting things to motor neurons. I've highlighted how important this is, and so getting things to Schwann cells, for example, is something we've not yet approached in humans. There was some encouraging data yesterday about looking at sort of AAV vectors throughout transduction throughout Schwann cells, very encouraging. Now we need to take that to people. I would say in terms of biomarkers, I think that the CMT field, there are some emerging biomarkers. You heard yesterday we heard about muscle fat fraction as a really exciting biomarker. But putting that all together and saying, okay, how does this relate to the clinical course is probably a next step. But fundamentally, what I'd end on is, I mean, I've had a journey myself of years ago working on CMT, now recently coming back to the field, and I'm incredibly optimistic to see where it's at. There's this excitement about the science, there's the emerging technical tools we can do to make therapies. People are developing the biochemical tools, the biomarkers, the clinical trial apparatus to really make a difference. And so I couldn't be more enthusiastic about where this field stands. With that, I'll close and take a question.
Thank you, Dr. Ferguson.